The Endosymbiotic Ratchet and Plastid Evolution. Plastid endosymbiosis explains the origin of the photosynthetic algae and plants that form the base of the food web in many aquatic and terrestrial ecosystems. Evolution of the plastid organelle in these species involved the capture and retention of a photosynthetic cyanobacterial cell by a eukaryotic protist about 1.6 billion years ago. The radiation of this lineage into the Archaeplastida, which includes green algae in plants, red algae, and glaucophyte algae, happened soon thereafter. This gave rise to 3 distinct lines of descent that share very few characteristics besides their ancestral plastid, which begs the question, what drove the rapid divergence and evolution of the Archaeplastida host cell, and why? These questions are hard to answer using plants and algae as models because of their ancient origins. Fortunately, the photosynthetic amoeba Pollinella offers an excellent alternative model system because its plastid originated only about 120 million years ago, and this novel algal lineage is still undergoing rapid evolution to allow its survival and spread around the world. We analyzed existing genome and morphological data from several Pollinella lineages and found that, even though they are young in evolutionary terms, they differ greatly from each other with respect to cell size and shape, and in particular, genome features. These genomes vary from about 700 million base pairs in size to almost 10 billion base pairs. This contrasts greatly with the heterotrophic ancestors of Pollinella that are much smaller cells, with relatively tiny genomes of about 20 to 40 million base pairs. These observations led us to propose the endosymbiotic ratchet hypothesis to explain the early evolution of photosynthetic lineages. We postulate that plastid origin creates a ratchet that drives lineage innovation, speciation, and rapid divergence. This likely happens due to a sharp reduction in effective population size, NE, in ancestral algal lineages, due in part to poor integration of the novel photosynthetic organelle, which results in a slow growth rate. A low NE weakens the strength of selection and results in a higher mutation rate and genome growth due to the expansion of repeats and gene families, all of which can lead to rapid adaptive evolution in surviving cells. We believe that this period of accelerated evolution drove speciation, promoting the origin of sister lineages occupying different habitats that underwent the ratchet independently, as is the case in Pollinella. The endosymbiotic ratchet provides a general framework for interpreting post-endosymbiosis lineage evolution that is driven by disruptive selection and demographic and population shifts. It may also explain the divergent evolution of the Archaeplastida. If the endosymbiotic ratchet operated in the ancestor of the Archaeplastida lineages as it did in Pollinella, then it is no surprise that the red, green, and glaucophyte algae are so different from each other. They may also have undergone a period of rapid divergent evolution during organelle integration. Each Archaeplastida algal lineage explored different light and nutrient environments to minimize competition and survive in small, isolated populations with low NE. However, once they adapted to their different environments and had well-integrated plastids, their evolutionary trajectory and potential habitats were widened, leading to the amazing diversity of algae and plants that we see today. The same may occur for Pollinella far into the future.